So, good evening and warm greetings to all. On behalf of Clinical Secretariat of Indian Pharmacological Society, it is my privilege to welcome you all to this special webinar on International Clinical Trials Day 2026. We know this year the theme is Stronger Together, Advanced Therapy Clinical Trials Without Borders. So, International Clinical Trials Day reminds us of a defining moment in medical science. We know 20 May 1747, when James Lind began his landmark clinical trials on sailors with scurvy, that simple but powerful experiment actually planted the seed of evidence-based medicine. Today, nearly 3 centuries later, we actually have moved from ships to satellites, from bedside observations to AI-enabled platforms, and from single country studies to truly borderless global collaboration. So for Bharat, this moment is especially promising with our scientific talent, digital health infrastructure, diverse population, expanding clinical research ecosystem, and growing regulatory maturity. India is not merely a participant in global clinical trials. Bharat is ready to become a trusted leader in ethical, inclusive, innovative, and patient-centered clinical research. So maybe we will get certain regulatory vision which could highlight a future of coordinated regulators, scientific and ethics boards, along with certain regulatory science centers and AI-enabled platforms. So we can engage patient, clinician, industry, and also academia. Today's program actually reflects exactly the future. We began with next generation advanced therapies like gene therapy, CAR-T, CRISPR, RNA therapeutics, and we expect insights on how these innovations can move responsibly from bench to global trials. We will also then explore decentralized and borderless clinical trials, including e-consent, remote monitoring, wearable biosensors, AI-driven recruitment, and also cross-border pharmacovigilance. So this could help our trials more accessible and inclusive. Our third talk will take us to the clinical pharmacology of cell and gene therapies when conventional PKPD thinking must evolve for living therapies, biomarkers, immune monitoring, dose optimization, and long-term patient safety. And then fourth session on regulatory harmonization will address FDA, EMA, CDSCO, ICH, and beyond, a crucial theme if advanced therapy trials are to become faster, safer, ethical, and globally acceptable. Finally, we'll look ahead to precision medicine, AI, digital twins, and model-informed drug development with real-world evidence, personalized therapeutics, and trial diversity. The future, the real future language of clinical trials. So I warmly welcome all delegates, faculty, students, researchers, clinicians, some regulators, industry colleagues, and all our friends of science. I also invite each one of you to become a member of our society, Indian Pharmacological Society, and join us in our commitment to science for all, science for equity. With gratitude, I request our two distinguished chairpersons, who needs no introduction, Professor Nilima Khirsagar Madam and Professor Shantanu Kumar Tripathi, two great clinical pharmacologists of immense national stature to kindly take over and chair today's webinar. Thank you all once again. Welcome to this ICTD 2026 webinar. Over to Professor Nilima Kirsagar, Madam.
Thank you very much, Dr. Sambhu, for that very nice introduction for this very important webinar on clinical trials, reminding us of the historical aspect and the importance of what things have happened since then, and highlighting for us the content of this webinar. We have some extremely experienced and knowledgeable speakers for this webinar, and I would therefore invite them one by one for this webinar. I don't know if we have any opportunity for audience to ask questions. There is a chat box there, so maybe they can put their questions in that. Dr. Sambhu has very rightly pointed out that India should be leaders and not anymore followers in the clinical trial scenario. We have now our diaspora in many different countries and some in very, very important positions in important leading organizations, and we are very proud of them all. One of the things that I would like to mention here, and I'm sure the speakers would bring up that, is the importance of diversity in countries like India, which are, while a good amount of development is happening in the Western world, there is always now great lead being taken in India itself for CAR T cells and such things. But this all brings us to the diversity of population that's going to use these products. And therefore, we often wonder about the importance of ethnic variation and such things, which need to be considered from the early stages of clinical trials and also from the early stages of product development. So it is considered that the various aspects of clinical trials, in particular, let's say the patient information and the informed consent, they are rather poorly, we all know they are rather poor translations of English patient information, as are also our package insert and such thing. So I think these are the areas in which now entire global community is looking at such aspects as we try to harmonize, as Shambhu rightly pointed out, various regulatory requirements. And the US FDA and EMA and everybody is now considering the patient focused outcome measures, which are crucially important in various clinical trials. I'm sure these will be touched upon by our excellent speakers. So I think our first speaker is Dr. Sanish Davis. It's my great pleasure to invite him. Again, he needs no introduction. He has been speaker at various forums, and he has excellent experience in industry. He's a wonderful speaker, and he's been the chairperson or president for the Indian Society for Clinical Research, and has guided that for very, very important conferences and activities. So I hand over the mic to Sanish and look forward to your presentation. Thank you so much.
Thank you, ma'am, and to the organizers for this opportunity. And if you can let me know if you can see my screen.
Yes, sir. Now it's
okay. Thank you. So at the outset, greetings of the International Clinical Trials Day. It's something for all of us who are working in research, whether we are as a site coordinator, whether we are an investigator, sub-investigator, whether we are in the ethics committee, whether we are in the regulatory agency, or whether we are just an innovator or just doing, you could say, monitoring from a sponsor side or a project manager. It is our day, right? And most importantly, the most important person in this whole enterprise is the patient. So everything that we do is for the patient. And this particular day is indeed, you could say, one of the biggest days that we can celebrate for, you could say, the hero of research, as we say, who is the patient. So these are my customary disclaimers for the talk. For the next 16 minutes, what we'll try to quickly cover is on patient centricity, the changes in, you could say, treatment paradigms that have occurred over the last, you could say, a decade in oncology and how this has changed. While I may not, you could say, touch on every different aspects of oncology, therapeutics, etc., I will focus my discussion more on immunotherapy agents, because this is by far the biggest, you could say, treatment class of agents which are now extensively used in countries like India as well as in other parts of the world. And our discussion will then go into what are some of the toxicity considerations, what are some of the clinical development consideration of these agents, because they have very specific toxicity profile. And then for those who are, you could say, from the clinical development side and postgraduates in pharmacology and medicine, most importantly, you could say the most important work is on, you could say, phase 1 trial designs that are there. And then we'll try to kind of like end up with safety, you could say, reporting of some of these studies, etc. Now, quickly, if we go through why patient centricity matters, most importantly, all of us agree that patients are partners to the whole enterprise. Patients, you could say, increasingly also in Western countries have also tried to kind of like influence trial designs as well as sometimes outcomes. It also ensures that there is greater relevance for the clinical trial, which ultimately leads to better patient outcomes and meaningful endpoints that are, you could say, captured in a clinical trial. Maybe quickly as an example, what I can actually showcase is that traditionally, I think progression-free survival was an endpoint which was utilized very commonly in oncology drug design. But now increasingly, I think overall survival is being looked at both by a regulatory agency as well as some of this thought process actually started with patients who said that I'm not bothered whether my cancer is, you could say, stopped to, you could say, increase in diameter or the tumor cell has increased, etc. What I'm more worried about is that how long will I survive? Because based on that, there are a lot of cascading or end stream decisions that happen. Like, for example, how long do I have before I can marry off my daughter? How long do I have before I can actually gather a corpus so that my family would not have to suffer? So there are a lot of decisions which actually come from better patient, you could say, decisions and ultimately meaningful endpoints in clinical trial. And then, of course, these kind of real world, you could say, experiences and perspectives from the patients also, you could say, tailors our future oncology clinical development. And it is also not, you could say, something that people do just for the sake that maybe a US FDA has put it in their regulation or ICMR has put it in their guidelines, etc. But there is huge value of patients' participation in clinical development. Like, for example, simple things like if more number of patient advocacy groups and patients actually are interested in following what is new, what is available for them, it can lead to accelerated recruitment in the clinical trial and also retention of participants in a clinical trial, which again means that the efficiency of clinical trial increases. And for a country like India, it also can mean more number of clinical trials actually come, which can serve our Indian patients in a big way. Regulatory confidence in the data increases. The drug development process become more streamlined because people who are sitting in the industry may not sometimes really understand what kind of patient perspectives need to be addressed. And the example that I said earlier is a classic example where overall survival becomes more important than progression-free survival in many ways. And then, of course, there are also advantages of having the product come to the market, which is a big win for any company who actually puts R&D money into this thing. So the oncology drug development as well as newer drugs that come are, you could say, the landscape is changing so fast. So, for example, something which was current or topical last year would now sometimes not even be relevant this year because there were new studies which came out, new results which came out and changed the entire landscape of that particular treatment. But three things to know: precision medicine is becoming more and more important, which is the holy grail of any treatment, which is personalized, tumor agnostic therapies, etc., which definitely has a big role for improving treatment outcomes. We are going more and more to individualized care with combination strategies more upfront rather than waiting for the drug to actually be looked at as a combination treatment many years into development. So a lot of these changes are all happening. And if you look at the current newer agents which are there on the horizon, as well as those which are already approved, so you have checkpoint inhibitors, a classic example of pembrolizumab, which is a PDL1 inhibitor, etc. CAR T therapy. Now, India has probably 3 CAR T programs and 3 CAR T agents which are there, which is, again, all these mechanisms are given on the slide, so I don't have to read it again and again. Antibody drug conjugates are something which is like a very intuitive way of combining an antibody with a toxic payload, which is like a drug, and then you inject it into the cancer cell, which is like targeted destruction. And then bispecific antibodies. You don't have just one antibody, you have 2 antibodies, one which has grabbing of maybe a T cell as its function, another which is like a linking agent. So both bring together complementary activities to kill the cancer cell, etc. So moving on, immunotherapy, and I will focus on the rest of the 10 minutes on this area. There are very specific toxicity profiles of these immunotherapy agents. One is called as cytotoxic release syndrome. So those who have been through COVID period would remember that COVID also creates a lot of these cytotoxic release syndrome, right? And that's where I think a lot of people actually brought it. It also produces neurotoxicity, which is called as ICANS. It's a very specific kind of neurotoxicity which is there. And these are very common sometimes in CAR T cells if they are very efficacious, etc. Needed to be treated very quickly, otherwise patients can actually have a respiratory failure and organ system damage and all that stuff. Tocilizumab, which we have used so many times during COVID period, is an agent which can easily be used to control this along with corticosteroid. And then you could say all this oncology development is, you could say, fired up by the regulatory agencies, you could say, pathways that are there. So, for example, breakthrough designation is a designation that the regulatory agency gives for drugs which are extremely different from what is already available as standard of care, or something which demonstrates substantial improvement over existing therapy on maybe an oncology related input. So if a drug gets this kind of a designation, sometimes the drug can even be approved just based on the phase 1 patient study which is done. And there are so many examples. Even in today's world, majority of the newer drug classes which are used in oncology actually has this kind of a definition. And this enables a lot of phase 1s which actually go on. And even in India, I think we'll have a lot of these things. But for immunotherapy agents, it kind of like turns on the principle, or it's like inverts the principle which is always followed for chemotherapy related drug phase 1 studies, which is that you want to actually get to what is called as the MTD, or the maximum tolerable dose, by finding the safety profile, which is like the finding out the DLTs and others. And that's what we call as the 3+3 design, that many of us would know in oncology drug development, where you try to find the DLTs and then move forward to find the maximum tolerated dose, etc. But for immunotherapy agents, this paradigm is so different, because the DLTs and MTDs are, you could say, something which is, you could say, very specific for cytotoxic agent. But these drugs have issues like late onset toxicities. So, for example, chemotherapy agents kind of like sometimes produce their toxicities in the very first cycle because they kill the cells, right? But in the case of immunotherapy agents, it doesn't happen that way, because it's not like directly toxic to the cancer cell. It is working more with the immune system and other things. So there could also be different immune related adverse events which occur outside, sometimes even cycle 1 or maybe cycle 2, etc. And the potential ways to deal with this is to, if you want to enhance the duration of the dose limiting toxicity window that you use, or sometimes use something like TIT CRM, which is a way to kind of like say that you're trying to assess at each cycle what happens to, you could say, the toxicity profile. Similarly, there is also a dichotomy between more is better and more is toxic kind of thing between efficacy and safety. So what this means is that when you actually increase the dose of the drug, the efficacy increases, but at the same time also toxicity increases. But for immunotherapy, this is not, you could say, a linear pathway, right? In the sense that the effects are not linear. If you increase the dose, it does not mean that more malignant cells are killed. In fact, it is going to be the reverse. So in immunotherapy kind of development, what you're trying to find out is the minimum effective dose, because you can't keep increasing the dose thinking that you'll get a, you could say, a similar higher efficacy or sometimes even toxicity which you are looking out for. But there could be plateau which actually happens. Similarly, the same thing when you move from phase 1 to phase 2, also what is called as the recommended phase 2 dose finding studies. Here also you will have to have a different strategy, because you can't just rely on one, you could say, endpoint, or even one DLT, or you find the which is that particular adverse event or serious adverse event which is defining your DLT. But you want to actually combine maybe an activity marker which is called as a clinical response marker to find what is ultimately called as the optimal biological active dose. So these are terminologies which are kind of coming into, you could say, oncology drug development, especially with immunotherapy agents, which you could say behave a little bit differently. And there is also the challenge of when you have multiple drugs which are getting used. I kind of like shift to my last part of the session, which is on safety reporting in these early trials, right? So, for example, many a times doing these studies are like a race in its own way, right? Because only limited set of countries are used, but every country wants to actually participate in this because there are huge benefit to the patients that is there. Even in there, we have been trying to kind of like get into many of these studies. Now we have CAR-T, so the technology is there. We also have the capability, but we need more of these indigenous drugs being developed so that our population can actually benefit from this in many ways. But at the same time, there are also these challenges. So I'm just kind of like giving us a snapshot of some of the challenges that sometimes are there. And these are sometimes pharmacovigilance related challenges which are there. So a patient who's on chemotherapy and an immunotherapy agent, say, for example, develops neutropenia, and the investigator feels that it is related to the study drug. And in there, we have to, the PI has to assess the relatedness. But then, to which drug is it? And there are pushback which happens from global organizations on these kind of this thing. One thing that can help a PI is to look at the investigator brochure closely, follow the literature that is there. And sometimes it is just that discussion with the sponsor medical monitor before you make the causality, because you can't keep changing the relatedness of the drug that is there. Sometimes investigators also do funny things. This is an example of one of the investigator in one of the studies actually talking about adverse events in a patient and what treatment to give uh, on X, or you could say one of the social media that is there. And you can see in one of the panels that the investigator is also asking what therapy would be best for the patient, like a survey that is there, which is also something which can be very dangerous in many ways. It is accepted that investigators do this in good faith, but ultimately disclosing the drug and the name of it is a breach of social contract. You could even have patients, they might sue the investigators if some of these kind of challenges are there. So reaching out responsibly by investigators is important. Similarly, sometimes reporting of adverse events can be a challenge. Like, for example, a headache. Headache is a very common occurrence in chemotherapy. But in an immunotherapy study, if this is not captured, and it is captured by in the nursing notes but not in the investigator's assessment, this can lead to challenges with regards to reporting that is there. Similarly, oncology studies are rife for having huge volumes of SUSAR reporting, etc. And sometimes it is up to the site to actually figure out how to actually let the EC or IRB make a good decision, because no ethics committee or others can actually go through these huge amount of you could say stuff to actually understand what is the action that needs to be taken. Maybe here is a work that the sponsors can do to make it much more better for India or other countries, etc. So to end up my session, immunotherapy is an established form of cancer therapy. They develop impressive clinical activity across several tumor types. The classical assumptions of drug development for cytotoxic agents may not hold applicable for immunotherapy, because you need to have differences of way to do this. Immunotherapy agents have its own safety reporting needs, and hence investigators need to act responsibly here. And there are specific approaches that all stakeholders can ensure to put in, so that patient safety and data quality can be of the highest quality. So thank you once again, and appreciate if any questions or comments from folks who are there on the call.
Thank you very much, Anish. Tambu, do you think there are any questions? Are there any questions in the chat, or anybody would like to ask any questions?
I think, madam, the question will be put in that chat box. Later, they will collect that question and that will be given in the chat box, madam.
Okay,
If you have some comments, or if you want to ask sir something, we can have that question and answer session. Otherwise, we can move towards the next session.
Yeah, I don't have any questions. I just want to congratulate Sanish for very fascinating new development, new field, which is has so many challenges, and I'm sure he has a wealth of experience in this area, and he has much more. I think he could have taken a one whole day's webinar himself on this topic, elaborating on each of the slides and extensively. I'm sure that is something. Maybe Sambhu, I could suggest you have someday one webinar just on the subject of immunotherapy for immuno-oncology, which would be, I think, a great learning for everybody. But thank you, Sanish. And I think we could give him a virtual clap for all the
Thank you. Thank you so much.
All the new information. Some who have done that, yes, we could do that. And then, yeah, the clap. Yes. We move on. That's so nice. We move on to the next one. Saket. Our Saket is here. Hello, Saket. How nice to see you.
Hello, ma'am. Hello, Dr. Sanish.
He is another fantastic speaker, and on many and working on exceedingly important areas, and it is in a global setup. So I think without further ado, I request Saket to go ahead. I don't know, Sambhu, whether you have a slide about the speakers' brief CV or something, because each one of them have achieved so much in their one or
Actually,
two
madam, we don't
decades
have the
of.
CV slide, but I request if the first slide of our presenters, if they have.
If they have put anything, yeah. Okay, Saket, please go ahead.
Sure, sure. Thank you. Thank you, ma'am,
Thank you.
and Shambho and team, organizing team. Uh, and Dr. Sanish has set a very nice context to the overall presentation and this whole series of uh, the talks on this important day, International Clinical Trial Day. We are all gathered to understand the nuances of what is happening new. And I have been given this topic by the organizing team about decentralized and borderless clinical trials and the new era of digital pharmacology. So I am Saket Thakur. I am currently working as a clinical development and project lead at CEPI, that stands for Coalition for Epidemic Preparedness Innovations. I'm based in UK, London. I'm basically overseeing the clinical development and end-to-end clinical and overall product development strategies for pandemic vaccines. These are basically targeting the WHO priority pathogens. I'm physician by training and DM in clinical pharmacology, and I'm KM alumnus and student of Shri Sagar ma'am and junior to Dr. Sanish. So it's again a esteemed group where I am presenting this. So in this talk, I'll be presenting hybrid DCTs, remote monitoring, e-consent, wearables, AI-assisted recruitment, how pharmacovigilance is shaping the safety into this borderless and decentralized clinical trials. So to define what this decentralized clinical trial stands, I would refer to the FDA's definition is about, so uh, the FDA's definitions on trial-related activities that should occur at locations other than traditional clinical trials. So essentially, what FDA emphasizes here is that these are the trials which are delivered through telehealth, local healthcare providers, involves remote data collection, and home-based assessments. However, you have to understand that a DCT is not fully virtually conducted trial or with lowered GCP and ethics standards or technology choices which made before endpoint and risk considerations. So essentially it focuses on the tenets of the clinical trial or participant safety, right, safety and well-being. Core design principles should be part of any decentralized clinical trials that mapping each activity individually before decentralizing it, retain investigator or sponsor accountability for delegated activities. The trial should ensure source data integrity, audit trail, and validated transfer following ALCOA principle, and pre-specify escalation pathways for clinical review like eligibility and endpoint adjudication, safety review, urgent care or emergency or safety management, and unblinding. These are the essential aspects of the protocols, and trials should specify all this into the framework. Why we need to decentralize trial, and COVID-19 could not be a better event in the history of clinical research that made us adopting decentralized in a wider manner. The first advantage is access, lower travel, reduced site burden, longitudinal data, near real-time symptoms through PROs, e-diaries, wearables, etc., improving adherence, representativeness, fewer geographic constraint. Now we can't say that we can't reach to the participants in remote locations because of digitization. Resilience, continuity through site disruption as we saw during COVID-19. So decentralized where access, burden, or data quality gains do not compromise safety oversight, endpoint validity, or inspection readiness. That's the core message while we decide to decentralize any trial. We see the spectrum. Same trial could be decentralized differently. For example, a complete, on the left-hand side, you see the site-centric model where everything happens at the site. And on the right side, you see complete remote heavy, like fully remote trials. And there are examples to fully remote trials as well. I'll show you how one of the examples of that. But having said that, the ideal mix would be something in between, like hybrid trial, where some intervention can happen in clinic, which are mandatory, required as part of the scientific integrity, whereas those which can ensure convenience to participant and can ensure or improve the data quality could be made remotely, and then combined hybrid model can be adopted. Having said that, the critical quality factors will still remain, as I mentioned earlier, like participant safety, for example, medical oversight, emergency pathways for management, AE escalation, unblinding, ALCOA principles, IP, which is very important in any clinical trial. For I'm working on vaccine trials and cold chain or any other trial for that matter. In fact, immunotherapy also requires a stringent temperature control. Any temperature control medicine should be following given in the right setting and at a right environment with proper monitoring and oversight. For example, which activities are to be performed by whom. It has to be documented appropriately. The digital systems where you are actually using these tools has to have vendor qualifications. There are standard regulatory guidelines on this, which has to be followed. System validation protocols, uh, then user acceptability test, UATs has to be included into any systems, digital systems, and centralized monitoring systems. Then QTLs has to be built into the remote monitoring systems. I'll talk about that in later. But essentially, the DCT has to stand the inspection test that can sponsor, reconstruct who did what, when, with source data, and for every clinical activity you can do it. Then you should consider decentralizing. What DCTs deliver on the left, where you can see what is real, and on the right you see risk of controls are absent. So on left, of course, it reduces participant burden, it improves retention, improves data flow and frequency, you avoid missing data, it's operationally resilient. But on the right, it also comes with its own biases. For example, selection bias. Digital only outreach exclude participants or parts of target population. That's an inherent bias. Differential ascertainment bias, uneven schedules and missingness across arm, whether it is missingness at random, not at random, and statistically how we can improve through sensitivity analysis. There are mechanisms to that, but still it is a risk. Information bias, unvalidated devices, unlocked algorithms, but that can be addressed through validated devices and locked algorithms. I'll talk to it how to lock the algorithm for digital biomarkers in subsequent slides. And of course, algorithm bias it's itself is a limitation, like subgroup level error patterns from the training data sets itself. These models, especially the AI training models, are very biased in terms of what is fed to them in a training data set. And if this training data set is biased, then probably the outcome that it comes is going to be biased. So garbage in, garbage out. So that is very important while you make the training data set and train your systems. And each element must reduce the burden without compromising representativeness, interpretability, or inspection readiness of the trial. Uh, I'll come to COVID-19 examples, and it kind of a flagship event in the history, as I see it. It didn't decentralize the pivotal trials, it decentralized part of them. So we had emergency guidance released during that time from FDA, EMA, MHRA that enabled remote consent, direct to participant IP delivery, telemedicines. So essentially, it managed all the trials during disruption. That was not deliberate DCT design. It mostly drove early adoption because of this flexibility approach. So few on the left hand side, some of the intervention in those COVID-19 DCTs were kept site based, and on the right they were decentralized. So intervention administration like vaccine, as I said, that was kept site based. trained healthcare professionals were administering. There was at least 15 minutes post-dose observation. Anaphylaxis kit were ready. Primary efficacy endpoint for all the trials was RT-PCR confirmed plus clinical and adjudication of symptomatic COVID-19. There was an investigator oversight on safety. There were SAE management protocols, AESI management protocols, and there was an emphasis on sponsor and PI accountability throughout. The component which were decentralized were recruitment, like online registries, EHR, pre-screening, community outreach, reactogenicity capture of vaccine through e-diaries for solicited local and systemic reactions, 7 days post-dose observations, and even the safety events, like even we are now receiving data on updated safety of COVID-19 vaccine that is contributed through various online apps like VSEF, VST, Linked EHR, we have seen in NHS-based studies, Zoe app, which was adapted in UK for symptom reporting and safety reporting. So essentially, this was a hybrid model, and that delivered regulatory grade evidence with operational resilience. And I think this kind of created a benchmark for future trials, especially for vaccines, I would say. What they actually delivered in practice, and you see the results. For example, the famous BioNTech Pfizer trial of vaccine reporting 95% efficacy. It randomized over 40,000 participants across 152 sites worldwide with 2 doses. And the DCT elements, as I told, was e-diary for solicited reactions. Site base was dosing, primary efficacy endpoint, and clinical oversight throughout. Recovery trial for dexamethasone that kind of changed the practice during COVID-19 was again a platform RCT that was done through run on 176 NHS sites in the UK, randomized dexamethasone versus usual standard of care, and kind of given an evidence to use dexamethasone in the practice. Another not vaccine based, not to be biased on vaccine, I would say Apple Heart study, a very wide study that enrolled over 4 lakhs participants through app over 8 months and over 117 days median monitoring. It delivered 0.5%, 52% not irregular pulse notification through app. So it's a plethysmography based uh, app which monitors your rhythm. And if it finds irregular rhythm based on the pre-fed algorithm, it will give you an alert. And those who receives alert will given an ECG patch. So it matches the rhythm between your watch versus patch and identifies what triggers atrial fibrillation. And interestingly, the positive predictive value for this notification was found to be 0.84, which is quite substantial. So essentially, these are the example where digitizations or decentralization actually delivered the results. But you have to understand that you have to define a problem and then decentralize. Decentralize alone is not a solution. So at the core of the decentralization has to be the problem and not the scale, I would say, on which it operates. Eventually, they reach the scale, but they cater to the defined problem essentially. E-diaries again is are the standard components of any trials for safety reporting, especially vaccines. So there are limitations we know, and there are advantages. Essentially, we need to have protocol checks and balance, like what participant report has to be graded and converted into credible evidence. Monitoring is not excluded from self-reporting or participant-based reporting. You need to have source data verification and participant training, digital literacy, compliance management built into the protocols and data evaluation. And as I told, that usability testing, training of sites and investigator and participant education remains at the core of any of these apps when we are deploying for clinical trials. E-consent, always widely talked about phenomena in digitization and very much now integral for decentralized and digital trials. but it doesn't escape a standard framework of any informed consent that we take, like starting from information, interaction, understanding, voluntariness, and documentation. Although the layers would change, for example, information is given both way, in person or digitized manner, layered. You have translations requirement. Interaction would be live, given time for questions, understanding through proper questionnaires, teaching back, giving feedback, and documenting voluntariness like refusal and withdrawal, and documentation through e-signature. So this is non-negotiable for any informed consent. For evidence, I would say you see a Malawi trial, which is done in LMIC settings for a pneumococcal vaccine for one of the controlled human infection pilot trial. And it's a very interesting design because they used comparative assessment for both digital and the conventional traditional consent. They digitized the data, the kit, but having said that, because it compared both this, they took a selection bias in sort of in terms of selection population, because they had to educate digital literate population for paired comparison, and then the data was kind of significant. So you see, 100% participant completed e-consent without documentation errors, and whereas the paper consent showed errors in almost 43% of the documents. So essentially, it was widely appreciated by the participants, but the limitation, as I say, that it was limited to only adults without audiovisual impairment or the digital literacy, whereas we need to make them friendly or adopt a hybrid approach into a trial, that those who are comfortable with e-consent can be done e-consent, or e-consent followed by paper consent, opt-in, opt-out approach like conventionally been discussed for e-consent has to be implemented for make them to be inspection ready. Remote monitoring is becoming sort of a norm for any of the trial, not even digital or decentralized, but with digital trials that comes with a lot of data. That doesn't mean that a lot of data with digital devices or digital tools require source data verifications or indiscriminate source data verification. Remote monitoring, we saw that is work on a standard principle like data relevance or risk-based monitoring approach, where it starts from source data review to uh, adapting into a data platform like validation checks, audit trails, transfer logs, query workflow. Then this goes to a centralized monitoring systems for sponsors, like they do a statistical outlier apply quality tolerance limits to those data. If they are beyond those quality trial limit, then query is going to investigator about correction of data or risk flags as a risk to the key data like endpoints, safety endpoints or efficacy endpoints, and requires intervention both from the sponsor and investigator. And as I told, that action could be site contact, participant review, DSMB meeting, or escalation pathways. Essentially, the principle is that remote source is not a license for indiscriminate STV. We need to monitor what materially affects participant safety, rights, and endpoint credibility. Again, digital biomarkers will become a norm. Having said that, these are not being endpoints, valid endpoint, they are still exploratory. I'll give you an example of atrial fibrillation screening, a nice data set giving you a very important data on the positive predictive value. Even for COVID-19, several apps gave a lot of data on heart rate variability, physiology variation after vaccination, like for this study, DETECT, where feedback cohort of more than 5,000 participants reported increased heart rate after Moderna vaccine, and that was more pronounced in younger participant. Similarly, for other devices, this were observed. But authors of this article and they actually concluded that these are still remaining as exploratory biomarkers. They cannot become a surrogate for immune response, and they require a long chain of validation. For example, it requires 4 steps for any digital endpoints to or signals to cross to the validation pathway. So first is verification of the device. That device captures and transfer the sensor reliably under all conditions. Then the analytical validation about accuracy, precision, clinical validation that it relates to a clinical outcome, severity, meaningful change, and operational validation. For example, wear time, missingness, alert handling, audit trail. So pathway for any digital biomarker to pivotal use as an endpoint would be as a defining the concept like standard endpoint definition, how participant field function survives, then selecting a measure like physiological measure, algorithm feeding to that sensor, then context of use, then validation through three steps, verification, analytical, and clinical, and then pre-specify like ICH E9 estimand framework, endpoint role, how missing data will be handled, sensitivity analysis, and interpretable evidence. So just to summarize, they are still exploratory in therapy areas like neurology, like capturing through actigraphy devices of mobility, tremors, and using in indications like Parkinson disease, MS, stroke. Cardiorespiratory, we know the devices, Fitbits, Apple Watch, etc. They track your monitoring. Dermatology, you have these wearables and photo like capturing the photographs and sending to the dermatologist for psoriasis and eczema. Infectious diseases, for vaccines, you have tolerability and reactogenicity assessment, but still there are limitations like free living reliability for neurology, false positive, false negative, for dermatology, construct validity, for flares, meaningful change difference. Uh, Essentially, it has to go through following match endpoint role to evidence. Then, once you establish all these four steps, as I told earlier, then engage regulators like adopting FDA's DHT pre-submission pathway, EMS novel methodology pathway that exists, and you can get regulatory concurrence early. Then lock the algorithm, as I told, that is very important. Like for changes, after lock will require all rework against going back to the same pathway. So algorithm changes is to be locked, then define endpoints based on those algorithm, establish a framework, and put it into a protocol. AI assisted recruitment is again becoming a very important, especially in the Western world where you have standard EHR systems where protocol criteria are fed into data sources. And then AI NLP models will rank the participant based on protocol criteria. Investigator will review and decide whether to enroll participant or not. It will certainly reduce the workload, but as I told that it comes with its bias, like sampling bias. Training data will draw the eligible participants to a more favorable algorithm preferred patients. So you need to have some benchmarks like eligible screened versus enrolled target to see whether the targets match. Measurement or coding bias. We need to see ICD definitions or protocol definitions are fed into the EHR systems. Access, like digital outreach favors connected populations, then how to address the non-connected. So you have to establish alternative routes for outreach. And as automation will be always be challenged unless you have a human intervention included at some measure. So human intervention will be always needed to confirm the eligibility. Now, PV has always been a forefront in terms of decentralized. We know that the PV evidence or safety risk benefits assessment is always decentralized. It relies on various streams of data to feed into the overall risk benefit assessment and feed into the signal assessment. So PV streams of data would come for active follow-up, like for protocol defined visits, through apps. I told VSafe, eDiaries, app or web-based reporting that gives you a near-time data and short-term health impact reporting. So, but the limitation is that it is limited to a particular setting or protocol, and you can't reach the entire population, especially during COVID-19 sort of emergencies where you have vaccines deployed in masses and data has to come from various sources. Then comes supplementary systems like spontaneous reports like VAERS, yellow card system in the UK, ultra vigilance in UK. In India, you have PVPI reporting. So these are spontaneous reports that gives the early detection of unusual or serious safety hypothesis, but that doesn't give you causality. We know that they don't have a denominator, so that comes as spontaneous report, but still they are credible data. What comes as a next link is connecting the data sets between the background systems and what is existing in the healthcare system, like VSD, EHR, claims database, where you have all the data, and then you can compare observed versus expected rate for any event and see the disproportionality. This actually generates the signal, this kind of disproportionality assessment, and that can work through evidence generation and quantifies the signal further. There are some very good examples like GVDN as a very famous study, and I'm kind of indirectly involved into this study through my work, that actually generated 13 adverse events of special interest after COVID-19 vaccination in over 99 million vaccinated individuals across 10 sites in 8 countries using an observed versus expected design. So what did these studies do is that the standard case definitions of Brighton collaboration for each AESIs were fed into the protocol, and this data actually was fed into various EHR systems or standard databases and compared the AESI against those case definitions and gave the standard data rate for like myocarditis, pericarditis with Pfizer or mRNA vaccines, or Guillain-Barré syndrome, anaphylaxis, thrombotic thrombocytopenic syndrome, or VTS with CHADOX. So all these kind of giving very important data through integration of these systems, and that contributes to on the right, that is signal generation and signal quantification and eventually risk benefit assessment. Multi-central DCTs comes with its own challenges like cross-border governance, local clinical trial accountability, HCP qualifications, IP cold chain, as I told, equitable participations. And for vaccine specific, I told everything, so no need to be so specific about it. So essentially, burden reduction on participant motivates decentralization. Safety critical activities require clinical oversight. So where digital pharmacology is heading? So adaptive platform trials with DCT elements will become the norm as we go along, like principal recovery, uh, uh, then COVID-19 sort of trials will become the norm. Federated learning for safety signal detections on risk benefit generation. We see convergence of regulators on digitization of endpoints, DCT guidance, statistical measures on handling the endpoints, digital endpoints, digital biomarker qualification, and AI augmented trial conduct. So key takeaways from this talk. First is DCT is an operating model. We need to map each activity individually and delegate where justified. COVID-19 sort of validated the model, and that will become the norm. E-consent, variables, and AI are process tools. They enable rigor. They don't create it actually. E-consent is requires the standard participant understanding and autonomy measures and inspectable evidence. Variable endpoint has to follow the standard V1 to V3 validation and those early regulatory engagement. Pharmacovigilance demands evidence integration. No single stream contributes to evidence. It has to be through all the integration approach. Of course, the new AI NLP models will enable and improve this further. Having said that, this requires uh, a critical signal validation and signal generation strategy for comprehensive benefit risk assessment. And lastly, borderless trial demand local accountability, as I told, because each geography has its own challenges, uh, HCP challenges, HCP training, participant understanding, digital literacy, access has to be managed. So essentially, where, as I told early, the core problem is the driving factor for digitalization or decentralization rather than the scale or the novelty of the technology which we want to adopt. So lastly, I would say digital pharmacology is credible when it improves the reliability, timeliness, or interpretability of clinical relevant evidence and ensures the safety and well-being of the participant. Thank you. I would end my presentation here and invite any questions or comments.
Thank you very much, Ankit, for that extensive coverage of recent clinical trial excellent coverage. I just have one question that bothers me that, when there are decentralized trials that being conducted all over the globe, now how does one overcome the language barriers, especially because now we have everything is so much digitized and you have all the codes for various things, but the patient expressions of their symptoms may be more than what's coded in. So are there any thoughts or any ideas as to how this is being covered?
Yeah, that's a very good question, ma'am. So in e-diaries, generally we include the translations, language translation, preferable standard language translation, essentially catering to the literacy and comfort level of the participants. So that is to be done for informed consent also. I'm not sure about e-consent, how translations are managed, but I would rely on the correct translation, information delivery, and documentation of the understanding as a correct. And as I told that it has to go through all the four steps of the validation, like what you want to digitize, define, then rope it into a concept, validate, revalidate, and then deploy.
Have you come across any specific issues where the language translation has become an issue and therefore you had to, sort of relook at, or you just put it aside that this is something which we can't handle? Just keep it aside. Has there been any such example or issues?
Not for the decentralized trial per se, ma'am, but yes, language. In my previous experience as an investigator, we saw some literal translation of some of the reactogenicity terms, where participant actually gets confused what to report and how to report, and there we have to give feedback to the sponsor that these literal translations doesn't make sense in terms of both informed consent or e-diaries, where you need to understand that it has to be culturally appropriate. It has to make sense from clinical standpoint. So yeah, there the clinical eye comes into the assessment.
Yeah, right. Thank you. Thank you so much. And there are some few questions in the chat. Is there any method of AI extrapolation of kinetic models? And I don't know, there is time. Shambhu, is there time for answering these questions?
Yes, madam, we can take 2 or 3 questions, madam. Then
Yeah,
we can
okay.
move forward to the next session.
Yeah, next speaker is Sandhya. Yeah, Saket, if you're able to see the questions, you
Yeah,
guys
yeah.
answer them.
Yeah, of course. I think that the next the question from Dr. Manisha is more relevant to Dr. Sandhya, but I can address from the simulation perspective. Yes, it's feasible to simulate a small trial, a data set to a larger trial. There are some companies that actually do that. We are kind of engaging with few of them to see if they can be done. So what they actually do is that they take out the chunk of the data set, they train their model on pre-specified outcomes, and then build on a larger trial. There are approaches to in silico simulate placebo or external control trials. Companies use digital twin models for simulating a control trial outcomes to compare with the intervention outcome. So in nutshell, yes, this is possible, but still has to go afar in terms of model evaluation, requirements, therapy area wise, because for oncology there is rich data set, but it doesn't fare similarly to vaccines where we have very sparse training data to be fed into those models. So where there is practically no data set to be used for such large scale simulations. So it comes with very differences across therapy areas. But yes, this is possible and it's soon going to evolve in this direction.
I think there is some issue with Madam Madam's connection. Can you hear
Okay,
us,
can you?
madam?
Yeah, I can hear you. Yes.
Yes,
So,
yes.
yeah, so if there are no questions for Saket, then we can invite the next speaker, Dr. Sandhya. Great pleasure and privilege to invite you.
And I am just
Yeah, thank you,
requesting.
ma'am.
I am just sorry to interrupt, madam. I am just requesting the Clarnet team and to post those question answers to the group also. May I request Dr. Saket sir, if you just type down few salient points to these answers, and
Yeah.
that can be given in our main that is delegates box, so that our
Yeah.
delegates will get those answers. So I am also getting another question. I am just typing that here. And
Okay, I'll do that.
may I request Thakir sir to please note
Yeah.
down those answers in a brief, and so that our Clarnet team will post that to the main group. Thank you.
Okay.
Thank you.
Sure.
Thank you.
Over to madam.
Thank you so much, Dr. Sandhya. Yours. It's just of time you know that introduction such a person Sandhya. So please go ahead.
Yeah, thank you, ma'am. It's nice to make a presentation under you. So can I share my presentation, ma'am? Can I start?
Yes, who is it? You can share.
Yes, madam. Please, please.
Yeah. Just a minute. Just 1 minute, I'll share my presentation. Is it visible?
Yes, madam. It is visible. Can you please make? Yes, it
Yeah,
is now
yeah,
full
yeah.
screen also.
Yeah. So like my network is little slow. So I would like to switch off my video and present. Is it okay? Or
Yes,
I should
madam.
just-
Sure. Yes, madam.
Yeah. Okay. Yeah. So I will just present with the video switched off, and once my talk is over, I will switch on the video. Yeah. So first of all, I would like to thank the organizers for giving me this opportunity to be a part of this webinar on this International Clinical Trials Day, and it is indeed a privilege and pleasure for me to make this presentation under Dr. Nilima Shitsagar, the great scientist of clinical pharmacology, whom all of us look as a great person and want to follow her footsteps. So with this, I would like to move on to my topic on clinical pharmacology of cell and gene therapies, PKPD beyond conventional drugs. So in the next 20 minutes, I will be giving an overview of the definition of cell gene therapies, the scope of cell gene therapy, and how it differs from conventional drug therapy, the unique PKPD principles of CGTs, dose optimization, biomarkers, and immune monitoring, long-term safety surveillance, and the summary. So to begin with, so long back, James Lynn, who is considered as the father of clinical trial, and based on whose achievement we are celebrating the International Clinical Trial Day on this day, that is 20th May, found it very easy to come out with a solution to treat seasickness with very few simple things, whichever was available to him at that time, like nutmeg, seawater, elixir, oranges and lemons, etc. And he was able to find out the positive finding within few days. But however, this is not the case as the clinical research progress. For various diseases like cancer, immunological diseases, genetic disorders, it's not possible to find a solution as easy as James Lynn found out. So if we look at the last decade, we have come across lots of Nobel laureates who have found various therapies to treat especially cancer, genetic disorders, and rare diseases using gene therapy, cell therapy, and mRNA, etc. So, our topic is going to be dealing with these discoveries. So, if you look at the recent advances in drug therapies, we have cell therapy, we have gene therapy, and RNA therapy. So, what is this cell therapy? Cell therapy is nothing but injecting cells to a patient to replace, repair, or supplement the existing cells or tissues to either treat a disease or prevent a disease. Most of the time, for this purpose, we use either stem cells or immune cells, and most of these cells are taken from the patient themselves, like it is autologous. Or we also have a gene therapy where the genes are introduced or the genes are modified in the patient so that it regulates, repairs, or replaces a particular genetic sequence and treats that particular disease. And the gene is introduced into the cell using either viral vectors like adenovirus, lentivirus, or non-viral vectors like nanoparticles. And we also have RNA therapies where the diseases can be prevented by modifying the gene expression or their cellular function. And we have mRNA. We came across mRNA vaccines during COVID. We have small intestine RNA treatment and antisense oligonucleotides. So these therapies, the gene therapy, cell therapy, and RNA therapy has created revolution in the field of medicine, and it has opened a new path for treating those diseases which were considered as untreatable once upon a time. Okay, so coming to my topic, cell and gene therapy. So what is this cell and gene therapy? It is nothing but a collection of gene, cell, and RNA therapies. These three therapies are collectively called as cell gene therapy. Cell and gene therapies. Gene, cell, and RNA therapies are together called as cell gene therapies. So instead of giving a separate cell therapy or gene therapy or RNA therapy, it collectively uses this. For example, a gene therapy just introduces a vector to produce some changes in a particular gene with or without modifying the cell. Whereas a cell therapy includes injecting a cell which will produce some changes in the host, or RNA therapies changes the gene expression. However, here these mechanisms can be used together, like the cell function can be modified by altering the gene within that cell. So collectively, when genes and RNAs are used, they is known as cell gene therapy. Okay, so what is the scope of cell gene therapy? Where the cell gene therapy can be used? This is mainly used in those diseases where the genetic cause is well known and characterized. In other words, only for those diseases where there is a specific genetic cause, this cell gene therapy can be used. So how they can be used? They are designed to correct that particular genetic cause which is causing the disease by delivering a genetic material to modify the expression of the target gene. Let us see this in the example which is coming in the next slide. So, for example, in spinal muscular atrophy, which is autosomal recessive neuromuscular disorder, this is mainly due to the deficiency of SMN protein. So, a drug, nusinersen, which is an antisense oligonucleotide therapy. If you look at the mechanism of this ASO, that is antisense oligonucleotide nusinersen, it is basically a single-stranded nucleic acid. It targets the intron 7 of this SMN protein and modifies the splicing. As a result, it increases the production of functional SMN protein, which in turn results in clinical benefit. So the spinal muscular atrophy is mainly due to the deficiency of SMN protein. This is corrected by giving an antisense oligonucleotide, which in turn increases the production of SMN protein, thus resulting in clinical benefit. So if you look at the current state of cell and gene therapy development, there has been great progress in drug discovery and development with respect to cell therapies, RNA therapies, gene modified cell therapies. If you look at the cell therapies and gene therapies over the last decade, there has been a tremendous increase in the development of these drugs. And if we come to the development of these therapy in each region, almost all the regions are mostly involved in developing the cell gene therapy, like America, Europe, Asia Pacific, everywhere it's going on. So this clearly shows that irrespective of the continents, everywhere the cell and gene therapy related trials are going on, and they are going to come out in a big way in the coming future. Okay, so if we look at the FDA approval, as of May 2026, nearly 49 cell gene therapies have been approved by FDA for nearly 31 indications. So if you look at this, we have some cell therapy like autologous CAR T cell, we have autologous TCR-T, autologous TL, etc. And there are still more like in addition to cell therapies, different gene therapies, RNA therapies are approved by FDA, and the list is big, which is not shown here. Okay, now coming to the cell and gene therapies, how they are different from conventional therapies, why we need to know about these cell and gene therapies. If you look at the difference, as compared to the conventional therapies, cell and gene therapies are either living cells or genetic materials which are inserted into the host to modify the disease process, as compared to conventional therapy where a small chemical molecule or biological material is given to treat the disease. So in conventional therapy, we try to treat the disease by giving some chemical or a biological molecule, whereas here we try to give a cell or gene therapies, or we try to change the gene expression to modify the disease process. And if you look at the mechanism of action, unlike the conventional therapy which acts through receptors, enzymes, ion channels, the cell and gene therapies alters the cellular or genetic function. Either it modifies the cell function, or it changes the gene, or it increases or decreases the gene expression, and accordingly it modifies the disease process. And again, when we come to the pharmacokinetic, most of the time the ADME principles are not applicable to cell and gene therapies, which will be looking at in the coming slides. And if you look at the pharmacodynamics, unlike the concentration response relationship in conventional therapy, the cell and gene therapies have a different pharmacodynamic effect, like with respect to cellular expansion. Their action is based on cellular expansion, transgene expression, persistence, etc. And how do we monitor the conventional therapy? It is by therapeutic drug monitoring. However, it's not so with respect to cell gene therapies. Here we have to look for cell persistence, vector shedding, immune monitoring, etc. Again, I will be covering this in the coming slides. Okay. Again, immunogenicity is a major factor in cell and gene therapies. There are, of course, safety concerns. The cytokine release syndrome, insertional mutagenesis, immune reactions are more common in the cell and gene therapies, which is really little threatening for the safety of the patient. So if you look at the example of cell and gene therapies, as I already said, it can be a stem cell therapy or CRISPR-based therapy, etc. Okay, now coming to the PK/PD principles of the cell gene therapy. Unlike the conventional therapy, these cell gene therapies can be most of the time one-time dosing, or like few dosings within a short interval or at a regular interval. So there are not much doses with respect to PG therapies. So most of the time with one dose itself, the disease may be treated or get cured, or sometimes we may have to give it at a regular interval, either for a short period or a long period. And they have complex mechanism of action, which we are going to look into in the following slides. And they also have biodistribution and shedding instead of absorption, distribution, metabolism, and excretion. So the pharmacokinetics of CGT is mostly cellular kinetics rather than plasma concentration. So the action is not dependent on the plasma concentration, whereas it depends on the cell kinetics. And it also the PK is also based on various phases like distribution, expansion, contraction, persistence, etc. And as far as cellular therapy is concerned, for example, in CAR T cells, after infusion, they can proliferate massively. So the pharmacokinetics needs to be considered based on the proliferation. Because of these reasons, the dose response is often non-linear, unlike the linear pharmacokinetics with the conventional drugs. Similarly, with respect to pharmacodynamics, here most of the time the endpoints are measured in terms of cell killing or immune activation. And apart from therapeutic efficacy, they can also cause serious adverse events like cytokine release syndrome, neurotoxicities, nephrotoxicity, etc., or even bleeding. And the duration of action can depend on many factors. Some of the cellular therapy can create memory T cells, and the treatment duration may vary accordingly. And again the pharmacodynamics is based on the biomarkers as well as cytokines which are involved in producing the response. Okay, so because of these unique PK/PD principles, it's always difficult to determine the dose for a cell gene therapy, because we cannot determine the clearance based on the conventional pharmacokinetic concepts or based on the intrinsic and extrinsic factors such as weight of the patient or other comorbidities or co-medications. It's very difficult to determine the dose based on the intrinsic and extrinsic factors. And most of the time, the conventional PK/PD model is also not useful in cell gene therapies to determine the dose. So what do we do? So we have to go for some dose optimization strategies for better safety as well as efficacy. And population pharmacokinetic modeling seems to be better in identifying the various covariates which can be used for determining the almost near useful dose for patients. Or use of adaptive Bayesian dosing also improves the individualized therapy. Sensitivity analysis with respect to transporters, enzymes, and receptors also provides some information. However, more than these parameters, ODE-based cellular PK modeling seems to be very useful in dose optimization strategies. So let us look at this ODE-based cellular PK modeling. So this is nothing but ordinary differential equations cellular PK modeling, where a mathematical equation is used for cellular PK modeling. This model simulates a nonlinear transport, metabolism, and binding kinetics, and it describes the dynamic mass transfer between the compartments, and it provides a facility to track uptake, distribution, metabolism, and efflux of drugs within a particular cell. In other words, this ODE-based cellular PK modeling translates the biochemical pathways into mathematical equations to predict how a drug behaves at a cellular level. So this plays a major role in dose optimization in cell gene therapies. Apart from that, to know whether this the dose which is given to the patients is really useful, whether it is in the therapeutic range, is it safe, we also have to do immune monitoring. So what is this immune monitoring? It's something like how we do therapeutic drug monitoring for conventional drugs. In case of cell gene therapies, we'll be doing immune monitoring. Here, we measure the individual's immune response over a period of time when the therapy is given. How the immune response during various phases. For example, after vaccination, what happens to the immune response? Whether it increases or decreases, remains the same, or if it increases, how long it remains in the same increased status. So all these factors can be determined using immune monitoring. So this gives us a idea how long the therapy is useful, or it is going to produce more adverse effects, etc. So if you look at this diagram, when we give a cell or gene therapy, there's a possibility that it can produce clinical improvement at a particular dose in some patients, or it may cause no clinical improvement. Similarly, this is with respect to efficacy. Similarly, with respect to adverse effects, it may cause mild adverse event in some patients, severe adverse event in some, or it may even cause severe toxicity in some patients. So the responses still varies. Just like how we say one size does not fit all in conventional therapy, even in cell gene therapies, though they are all personalized therapy, sometimes the responses to the same intervention can vary across different people over a period of time. So to know this, we need to do immune monitoring. And how do we do immune monitoring? We have got various assays to do immune monitoring. For example, for cells, we can do like chemiluminescence assays, or we can do functional assays, cytotoxicity, etc. We can also measure the level of cytokines secreted. We can measure the protein levels using mass spectrometry. In case of RNA-based therapies, we can also measure the cytokines, etc. And immunohistochemistry, antigen expression, cancer topology, etc. can be done with respect to treatments where proteins are used. And in case of RNA therapy, we can also do sequencing to see if there are any mutations, transcriptional profiles, microbiome, etc. So this immune monitoring needs to be done in a lab which follows good laboratory practice, and it needs to have different equipments to meet the requirement. Based on the cell gene therapies, we may be requiring different set of equipments to measure the these levels, whatever is required for the particular study. Okay, so if you look at this, how this immune monitoring is helpful in this study. So where the patients were given anti-PD-1 therapy, the biomarkers, those biomarkers which were increasing with this anti-PD-1 therapy were seen by doing this immune monitoring. If you look at the pre-treatment and post-treatment, the biomarker which increases after giving anti-PD-1 therapy has been detected, and so based on this, so the percentage of responders from each patient group were assessed, and this particular biomarker was used as a biomarker to see increased drug response or better drug response. So just like how we say for conventional drugs, we have to give the right drug to the right patient with the right dose. In case of cell gene therapy, again, we have to give the right therapy to the right disease and to the right patient. What is the right therapy? We have to choose whether it is a cell therapy or gene therapy or RNA therapy or a combination of these three based on the disease. What type of disease? Whether it is having a specific genetic cause which can be treated by giving these therapies, and it needs to be given to the right patient considering the safety profile in that particular patient. So as far as FDA approval is concerned, FDA has been giving approval for lots of cell gene therapies, and some of the safety considerations which they are asking to look into includes shedding and insertional mutagenesis studies. So shedding is for especially for genetic therapy where the viral vectors are used for gene therapy, and this shedding is mainly with respect to community safety. Like the patient who has received the gene therapy may be shedding those virus into the community, which may be dangerous for the community. So before approval, shedding studies needs to be done. Similarly, insertional mutagenesis, like whether the the vector which is used for inserting the gene into the host gene cause mutagenesis, whether it binds to the wrong site in the gene or it overstimulates the gene and converts it into an oncogene, or it reduces the tumor suppressor gene in the cells. All those things need to be done by this insertional mutagenesis study. Similarly, the immunogenicity assessment has to be done, and a long-term follow-up is required for these type of cell gene therapies to ensure the safety, and it can be followed up even till 15 years. That's what the FDA guidelines says. However, the regulatory guidelines is going to be covered by the next speaker, so I didn't go much into the details of regulatory guidelines. So to summarize, the gene, cell, and RNA therapies are collectively called as cell gene therapies, and they have to be used mainly in specific diseases where it is due to a specific genetic cause, and the genetic cause can be treated. And cell gene therapies differ from conventional therapies in many aspects, including the PK/PD, which makes it difficult to determine the dose as well as monitor the drug response in these patients. So many techniques like PK/PD modelings are being developed to determine the dose, and immune monitoring are being developed to find out the safety of these drugs using biomarkers.
Thank you very much, Sandhya. Thank you for excellent coverage of this advancing and important topic. I think there could be some questions in the chat box, but I think in interest of time, we can maybe if you can see any specific questions, Sandhya, in the chat box.
I have not seen my
Not yet, madam. I think these questions are related to the last lecture.
Okay.
So
that
again,
Saket will be able to take up. So we can move on to Meenakshi's talk now.
Yes, madam.
Is she there? Has she joined it?
Yes, madam. Dr. Meenakshi.
Meenakshi, over to you then.
Sandhya, madam, can you please stop screen sharing, madam?
Yes, I am stopping, Dr. Shambhu. Yeah, I stopped.
Yeah, so may I now request Dr. Meenakshi Minu, madam. Madam is a clinical pharmacologist and associate professor at AIIMS, Bilashpur. So, madam, please, can you hear us? Madam, can you hear us?
So you're audible.
Yeah, I think Dr. Meenakshi madam is here, but can you hear us, madam?
Sir,
Hello?
yes, sir. I'm
Yes.
audible?
Yes, now you're audible, madam.
You can share her slides.
Yes, ma'am.
Yeah.
Is the slides visible?
Yes, madam, but you have to make it full screen. Yeah.
Is it now?
Yes, now it's full screen.
Okay. Good evening, experts. Thank you for giving me this opportunity to speak on global clinical trials, the architecture of regulatory convergence. Okay. As per my understanding, it's a lecture series by IPS, and I'll be basically talking about how the regulatory status of a static harmonization to now it is moving towards dynamic interoperability. Sir, is my slides moving? Yeah.
Yes, moving one.
So the overview of lecture is part one is core regulatory pillars, FDA, EMA, CDSCO, and ICH framework. Part two is emerging regional giants. AMP, that's African Medicine Agency, NMPA by China, and PMDA by Japan. These are the new voices in global trials. The paradigm shift which has been introduced by ICH E6R3 regulation, moving from process to proportionality. Then the dynamic interoperability, living dossiers, the RAS system, and the streaming regulation. There are three major pillars of digital convergence, which I will be covering in part 5. Basically, Reliance, Electronic CTD 2.0, and BASIN bridging. Finally, the critical challenges and outlook covering data sovereignty, quality maturity, and the key references. As we already know, there are few major core pillars of, sir, kindly excuse me, I have some cough too, which is FDA, EMA, CDSCO, and ICH. Okay. Now the global clinical trial landscape, what is the landscape in 2026? As I already said, we are moving from static approvals to dynamic interoperability. The 2026 landscape is defined by shift from static sequential approvals by different regulatory agencies to a dynamic concurrent interoperability, which is no longer an aspirational thought. This convergence is now operational and is powered by the massive advances seen in artificial intelligence and machine learning. Now, the ICH is basically the organization which provides the alphabet, harmonizing the guidelines and shared standards across all over the world. FDA, EMA, and CDS. It helps us in writing a unified language through real data sharing in cloud-based platforms for concurrent review. Now, I'll take a case in example, which is aducanumab. It's a drug which was approved by FDA under accelerated approval program, marketed as AduHelm, and in some time back, in this case, the primary endpoint was not the neurocognitive benefit, rather a surrogate endpoint was used. Two major pivotal trials were conducted. The result for one was positive, while the other gave a negative result. So overall, the result was inconclusive. Despite that, FDA granted a temporary license under this program. And you should know that the cost is around $56,000 for one patient. Like it has to be continuous infusion the patient has to take. I mean, the regulatory indication, approved indication is quite a monetary benefit to the company, Biogen. Okay, but because of that, the EMA, the application was withdrawn because the result is inconclusive. Okay, UK has a very struggling basically to reconcile the uncertain clinical efficacy with the cost of treatment. So with this example, what we say is different regulatory agencies having a different approach, and the outcome is not very, you can say, favorable in terms of either efficacy or safety or cost to the patients of Alzheimer's disease. So this way, now the FDA is moving from demonstrating a substantial evidence of effectiveness with one adequate and well-controlled clinical investigation, providing confirmatory evidence. This is also known as a plausible mechanism frontier, wherein the FDA now formally accepts one adequate and well-controlled study combined with confirmatory evidence through real-world data or a mechanistic linkage study as a baseline for marketing authorization. Simultaneously, FDA has also introduced something known as diversity action plans under FDORA 3601. It makes planning mandatory for phase 3 or pivotal studies, ensuring global trial data which should be demographically applicable to the US population. So moving beyond now the two pivotal trials, which is considered gold standard, towards total evidence framework, including in silico modeling and digital twins, to one study. Okay, there are two major projects which has been introduced by FDA. I'm just taking few examples where it is like more relevant. What are the regulatory shift or the framework? One is in FDA Oncology Center of Excellence, OCE. It has initiated a project Orbis in 2019. It provides a framework for concurrent submission and review of oncology products among the international partners. Okay. This allows the patients with cancer to receive earlier access to products in other countries, where if there is a significant delay in regulatory submission, and despite whether the product has received FDA approval or not. One example. Another example is Project Optimus. The Oncology Center of Excellence, the Project Optimus, is an initiative to reform the dose optimization and dose selection paradigm in oncology drug development. We know till now the dose selection was based on cytotoxic chemotherapeutics, but now in oncology we are moving from cytotoxic chemotherapeutics to the precision oncology, and using earlier design was actually not which was inadequately characterized before initiating the registration trial. So this massive project has been launched by FDA. These are the two major examples for changing the regulatory framework for oncology drugs. Okay, this is the same thing. Now, other point in Project Orbis is that first thing that it's a multi-country oncology review. There is more than 8+ international partners, including UK, Brazil, and Singapore. So we can see there is a convergence of the regulatory framework at one platform. The type A, it includes the active reviewers, and the type B is silent observers. So there are multiple types of reviewers in which the individual countries can actually become a part of regulatory review, that is known as the type A, or they become a silent observer. They can be a silent observers directly introducing the new drug which is approved by other regulatory agencies, for in which they were just observing silently how the regulatory process is going on, and they can directly into that drug can be directly introduced into their market. Now, this is based on a shared scientific consensus for an immediate market access. The Project Optimus, as I already told, it is a dose optimization revolution, and it prevents the costly late-stage failures, quantifying the total evidence and accepting the non-animal models. And this way, the FDA Modernization Act 2.0 is formalizing the in silico modeling and digital twins, which was also covered by Dr. Saket Rickerson. Okay. Now, the EU has focused more on the real-world evidence powerhouse, introducing Darwin EU, ACT EU and CTIS, and pediatric alignment. Darwin EU is one of the most advanced real-world evidence infrastructure globally, enabling rapid cycle studies on European healthcare databases to validate safety and signals in real time. While ACT EU and CTIS, it streamlined CTIS into a single entry point for multi-state trials, reducing administrative fragmentation across the EU. Pediatric alignment 2026 framework mandates the early stage assessment for drugs which is relevant to the childhood cancers, aligning with the US FDA RA requirement. EMA has further introduced some act for maturation. One is clinical trials regulation, CTRI number. It is now fully operational across all EU member states. So SNSA provides consolidated European perspective before the CTIS submission, eliminating the need for sponsors to navigate 27 separate national requirements. There is one single platform. Darwin EU network, is already told, is one of the most advanced real-world evidence infrastructure globally. And these things which I have already covered, but now it helps is integrating the real-world evidence directly into their full regulatory life cycle, which is also one of the goals for FDA. Okay. Now, where is India? India is moving from compliance to convergence very rapidly. Now, the ICH E6 or R3, it is adopted, which incorporates a fully implemented risk-based quality management in addition to the earlier risk band management. Now it's a risk-based quality management from the beginning of the study till end. Okay, it has introduced a bridging study flexibility. The new guidelines allows the exemption for drugs which addresses significant unmet needs with sufficient Asian Indian subpopulation in global trials. And we know it says that as for the new recent information, there is no need to conduct separate clinical trials for India if the global clinical trial has an Indian or as Asian subpopulation. Now, SIGAM portal has basically digitally transformed and supports the electronic clinical trial document submissions, enabling the reliance pathway, leveraging all the other regulatory reviews. The rule 101, and in this pathway at the same time, the drugs which is approved by assessment agencies can waive the local phase 3 in favor of the phase 4 surveillance. So if you can skip the phase 3, get the marketing approval, but then you have to show a phase 4 surveillance data. Now, see, in addition to those, CDSCO has also tried to bring about a transformation in their ethics committee by centralizing the entire ethics committee. Mandatory registration and accreditation of ethics committee via SUGAM portal. It has reduced the ethical variability. We know that companies they have to do a multi-centric trials, and every center has their own regulatory, own ethical review process, which is quite time taking. I have experience here itself. Now the NDCT from NDCT rules 2019 onwards, it is iterability refined to align with the ICH E6R3 guidelines, transitioning from a low-cost trial destination. Right? India wants to become in addition to generic hub, it wants to become a regulatory hub as well, with a mature reliance model leveraging the assessment agency decisions. Now, the emerging regional giants, we have AMA, which is African Medicine Agency, giving a single window oversight the entire African. All the countries are incorporated in a single platform, and they unified entry point for any clinical trial across the entire African continent, and giving a health sovereignty, which is, we all want to be a sovereign country, and data sovereign, will sovereignty, etc. So harmonizing the regulatory requirements to account for the Africa's unique genetic diversity, where there is a lot of problems there. Okay, so NMPA China and PMDA Japan. Okay, China is leading in AI, so there's no doubt they're also leader in Bayesian trial methodology, which is quite complicated methodology for incorporating the statistical and machine learning algorithm. But what they do, as with their other method, there is an external information borrowing. They don't hesitate from not copying, but rather stealing, which was also Steve Jobs' idea. So sponsors can use the global data more aggressively for Chinese approvals, and they're reducing the need for China-only replication studies. PMDA by Japan now, they are also waiving mandatory Japanese-only phase 1 studies. So if the data have foreign data showing comparable safety and PK profiles, though that is the condition, so they can waive the mandatory phase 1 studies. Japan is now trying to move into the first wave of global launches. Now the ICH E6R3, which is the digital constitution for clinical trials. From how it is moving from process to proportionality, it was adopted in late 2025 or early 2026. Now we have the risk-based quality management, which not one size fits all, moving from that to, okay, you decouple the trial conduct from the beginning itself and the data source. Now the quality by design, you build the quality into the protocol from inception rather than inspecting it for it at study completion. Now decentralized trials, it recognizes remote monitoring, e-consent, wearable data from wearable digital health technology as the primary data sources, which could be challenging in some countries, of course. Now ICH M11 is the protocol template, which is trying to harmonize all the documents. It is not just a document, it's a technical specification. The electronic protocol format enables the automated data mapping and works across global regulatory gateways seamlessly. Now ICH E21, the major point is about the regulators and ethics committee. It addresses the historically fragmented regulator-EC relationship. Now this fragmentation has delayed, I already mentioned the global startup time by at least 4 to 6 months on an average. This ICH guidelines aims to harmonize the parallel pathway of scientific and ethical review should go ahead parallelly. Now, how we have reached a dynamic interoperability by incorporating shared infrastructure and not just shared rules. Okay, how is it possible? Two things: living dossier, cloud-based reliance system. This is known as accumulus synergy. This is the first dossier in the cloud. The sponsor can host a secure cloud-based data room. Any regulator, whether it's FDA, EMA, CDSCO, they can log in simultaneously to review the raw data in real time. Okay, so submission gap up to 6 to 11 months submission gap is eliminated between US launch and the global launch. That we know earlier it was 3 to 4 years at least. Now, rolling review 2.0 was introduced. Now, the data is being audited as it's cleaned at the site level itself. Regulators can receive a secure token to access the real-time data lake, not some very, very heavy page document, which will take months to review itself. RAS, another process which has been introduced, is also known as regulatory as a service. The smaller nations can subscribe to the review expertise of major agencies. It is close like the LLMs. So we have not developed our own LLM. We subscribe to the LLMs which has been developed by large language models like ChatGPT or Gemini, etc., which has been developed by the US. The same thing here, the same idea has been transported here also. Now the smaller nations, since they do not have their own capacity to do this entire review work, regulatory review work, so what they can do is they can subscribe to this review expertise of major agencies, and this will prevent them or reduce their duplicated effort while maintaining sovereignty through tiered participation. The thing is because of the real-time data sharing and cloud-based data platform which is available. As I already told, there are two types of reviewers. One is active reviewers. The smaller nations can become an active reviewer, or they can just become a silent observer. Now, the end of the final report. So before, it was a snapshot model. You have very lengthy documents, static approval, etc. Okay, and a regulatory review begins only after the trial completion. Now we are moving kind of a continuous movie model. The automated feed of anonymized safety and efficacy signals is then we can directly take it from electronic health recorder from the trial sites. The issues can be flagged and corrected in weeks, and from day one there is a continuous oversight, all because of the advances in AI and ML. Okay, so major feature is 2010 it was a legacy model. Now we are in 2026-27, we have coming to a harmonized model. Okay, data focus was on static clinical study reports. Now we have moved to dynamic living dossiers. The logic for the review was sequential. Now it is concurrent. Trial design was major. It was large fixed RCTs. Now we are moving towards both single arm studies, real world evidence, adaptive trial design, etc. Octopus trial design, platform, or etc. The pillar guideline is ICH 6R2, which was process heavy. Now R3 is risk based and digital. Infrastructure, it was regional construction of the data, building regional silos. Now we have a multinational review platforms. So there are three major pillars of digital convergence, and what do we think how the agency is going to interact by 2027? There is a build-up of an access consortium blueprint. So Australia, Canada, Singapore, Switzerland, the UK, they have pioneered the single review multi-jurisdiction model. So instead of 5 agencies doing 5 reviews, one leads on safety, another one lead on efficacy, one will rely on the reports, and overall they will have a shared consensus of opinion on whether or not to approve this particular drug. The dossier as a service, this thing it helps in moving from a static snapshot to the data stream. The current ECTD 3.2/40 is still a static snapshot. So future is sponsor piloted data stream submission. Regulators will receive a secure token to access the real-time data lake. Data can be audited at discrete at the site level itself. Okay, and in the rolling review 2.0. Okay, in silico bridging a statistical solution. Okay, we know the local bridging study, for example, Japan and India, they require local data. Again, it's remain a major hurdle in global trials, adding cost and delays to every multi-regional program, which is India CDSCO is trying to avoid altogether. Even we have heard, I mean, recently that CDSCO is waiving all clinical trials in India. I don't know how much that is true. I mean, what is that? At what level that particular information is? So the solution is based in hierarchical modeling to borrow strength from global population and satisfy the local regulatory requirement. This of course will involve heavy statistical methodology. It's going to be really complex. Any Bayesian stats is really difficult. So expected by 2026-27 is formal ICS guidance on statistical bridging of regional population. They are going to release it using the global prior distributions to reduce local sample sizes, transform the bridging from a barrier into statistical exercise. Still, we have some critical challenges and the outlook. First thing is data sovereignty versus the data sharing. Now, the entire concept of this global convergence of the regulatory reviews rest on one thing, that there should be data sharing. But GDPR, European Union's GDPR imposes a strict data localization. Again, the personal information protection regulation restricts the cross-border transfer. FDA, though, demands an open data transparency, which data is being shared. And these three regimes are fundamentally at odds. So we rely on data sharing, but at the same time, there's a lot of restrictions have been imposed on data sharing itself. Okay, so the quality maturity gap, what is that? So regulations harmonize and inspection standards are not. GCP compliant sites, they vary wildly in practice, that we know. Essential documents is defined differently across regions, and closing this gap will require not only just guidance but training investment. Okay, these are my references, and I'm open to the questions by the experts. Thank you so much for listening.
Thank you very much, Meenakshi.
Thank you so much, ma'am.
Shambhuji will move on to the next speaker right away. We are already 20 minutes behind schedule. Okay.
Yeah, madam. But actually, what happened? Dr. Praful Mohan had sent one message because of their some army protocol and some emergency due to the death of the Uttarakhand chief minister, the previous ex-chief minister, I think Dr. Khanduri, I think. For that, they are now being involved in some process and procedures like that.
So he's not able to join.
Unable to join. So.
Okay, good. Okay. So I don't know if Dr. Santanu has been able to join.
He had some network issues, and that is why he had called me, because he is in Nepal now. He thought he can join, but some network issue for that.
Not able to join. Okay. So I leave it to you to summarize then, Sambhu.
Great. First of all, I want to thank you, madam, for your time and your valuable comments during throughout the session. And we are really thankful to all the faculties, starting from Dr. Sanis Davis, sir, who had started today's show. And then we had a great talk by Dr. Saket Thakur. And then we learned from Professor Sandhya Selvarajan, madam. And finally, we have a very informative talk and very comprehensive, we can say, talk from Professor Meenakshi Minu, madam. So really thankful to all the faculties. And last update was that we had almost 545 delegates who were viewing us live. 15 minutes back, I had got that update. So I am really thankful to all the delegates who were watching us. And on behalf of Indian Pharmacological Society, I want to thank you all for being with us. And especially there is comprehensive talk. I think we had learned a lot from all the speakers, and we will. Excellent. So
excellent presentation.
May I invite Professor Nilima Khirshagar, madam, to have a few take-home points for all of us on these topics. What is your take, madam?
I think these topics have been covered so extensively and so well that there is not much that I would like to speak on these topics, except sharing one or two, while all these advances are happening, there are some very basic things when it comes to clinical trials, and especially in our country. Unfortunately, some of these basic things like blinding and randomization and proper recording, proper carrying out GCP, source data verification, these things are somehow not properly attended to. And I would say that while we do want to have our countries advancing into all these new areas, we need to pay attention to these basic points. And in addition to all these technology and science advances, at the same time, I think the ethnic variation, culture, and language, communication related issues, and also proper consenting, these are the issues which basics of clinical trials we should not lose sight of these. But congratulations to Sambhu and Sanish and Meenakshi and Saket and Sandhya for excellent talk. And good night to all of you, and thank you all very much.
Thank you, madam. We have with us our president. We are really honored to have president of Indian Pharmacological Society, Professor Babul Bejborua, sir. So welcome, sir. So may I invite you to have your concluding remarks also. Sir, you have to unmute yourself.
It's okay?
Yes, sir. Yeah. Now you're audible, sir.
Thank you, Sambhu. And very good evening to all the respected chairperson, distinguished speakers, members of the Indian Pharmacological Society, delegates, students, researchers, and friends. Uh, as we come to the close of this important webinar on International Clinical Trials Day 2026, I feel both proud and hopeful. You know, the International Clinical Trial Day reminds us of that 28th May 1747 when James Lind conducted his historic trial on sailors with scurvy. Uh, and that moment showed the world that careful observation, comparison, and evidence can save lives. Today, almost after 280 years, the spirit remains the same, but the science has become far more powerful. So, this evening, the theme, The Stronger Together, Advanced Therapy Clinical Trials Without Borders, is truly timely. We have discussed the future of gene therapy, CAR-T, CRISPR, RNA therapeutics, decentralized trials, digital pharmacology, cell and gene therapy, PKPD, then regulatory harmonization, AI, precision medicine, and real world evidence. These are not distant ideas. They are already shaping the next generation of clinical research and patient care. For pharmacologists, this is a moment of responsibility. Clinical trials are not only about protocols, approvals, and publications, they are about people. They are about safety, ethics, rational therapeutics, scientific validity, and public trust. The clinical pharmacologist must remain at the center of this journey, designing better trials, selecting meaningful endpoints, understanding those response, protecting participants, interpreting evidence, supporting regulators, and ensuring that innovation reaches patient safety and equitability. Bharat, that is India, has a promising role to play with our clinical diversity, scientific manpower, digital health capacity, growing regulatory ecosystem, and commitment to affordable healthcare. India can become a global partner and leader in ethical, inclusive, and high quality clinical trials. The emerging vision of stronger regulatory science, AI enabled platforms, public dashboards, and active engagement of patients, clinicians, industry, and academia shows the direction in which we must move. So, on behalf of the Indian Pharmacological Society, I warmly invite all your young pharmacologists, clinicians, researchers, postgraduate students, academicians, and industry colleagues to join the IPS community. IPS is not just an association, it is a platform for learning, mentorship, collaboration, leadership, and service to society. Let us work together for science for all, science for equity. And now I sincerely thanks our distinguished chairperson Nilima Sirsekar madam and my guru Santanu sir for guiding today's deliberation with wisdom and grace. I thank all our eminent speakers for sharing their expertise and vision. I also congratulate the Clinical Secretariat of IPS for organizing such a meaningful program on an important day. Let us leave today with one clear message, that is, the future of clinical trials must be innovative, ethical, inclusive, and borderless, and pharmacologists must help lead that in future. Thank you all. Jai Hind.
Thank you very much, sir. So I think we are at the end of our session. As we had missed uh, at the last moment, Professor uh, Brigadier Praful Mohan, sir. So when I just came to know his absence, maybe 2 hours back, I had started some workup on that topic so that our viewers who are watching us today should get certain ideas about that topic too. So just I want to share that weekly, because we are all already at the end of today's session. Hope my screen is visible. And
Yes, sir, it is visible.
okay. So the last topic was this precision medicine, AI, and real world evidence. So AI is in the middle, and I had taken the help of AI only. I had put down 10 points, and I had asked my AI friend, AI colleague, to prepare one good looking slide. So it had produced this. So what is about precision medicine, AI, and real world evidence? If we start with this AI assisted trial design, there are certain AI models available which can identify the population, endpoints, risk, and adaptive pathways to minimize those risks. Then we have digital twin platform. That means certain virtual patient models to predict response, reduce the size, and therefore improve the safety. Then another issue is that the model informed development. I think we had a great talk from Professor Sandhya Selvarajan Madam. She had taught us about certain important PKPD modeling. But this type of model informed development could help us in simulation and dose optimization in real time with the help of this PKPD model. And then real world evidence means we have registries, electronic health records, post-marketing data complement, randomized trials. So we could have certain pragmatic trial designs. And when we talk about personalized therapeutics, I think we had a very great talk, the first talk by Dr. Sanis Davis. And we had tried to learn about biomarkers and biomarker-driven treatment selection. That is now the goal, and it is beyond that one-size-fits-all approach. And we need to acknowledge the Indian trial diversity. And in her last comment, Professor Nilima Kishsagar Madam had emphasized the role of ethnic diversity in India. And we need to understand this diversity, how it could be represented in terms of genetics, geography, gender, age, dietary practice, comorbidity, and inequity, the healthcare access. The equitable access is not there in different parts. So how that also can be managed. And the recruitment, when we talk about the AI recruitment, that means how we can find eligible patients faster while preventing bias and digital exclusion. We can detect certain rare adverse events, long-term risk, and subgroup-specific safety signals earlier. So safety via real-world scenarios. And I think we had a very great talk by Dr. Saket Thakur, who had told us about pharmacovigilance, how it could be, how we could reach beyond our geographical boundaries, regulatory acceptabilities, how the AI endpoints, their standards can be set, what are the evidence behind that AI endpoints, digital biomarkers, and how we could take real-world evidence decisions. I think the regulatory acceptability is an important part here also. And we had a great talk by Dr. Meenakshi Midu Madam on this aspect of regulatory science. And very importantly, our president, sir, also had emphasized the role of clinical pharmacologists, how their expertise could be utilized for lead dose individualization, benefit risk ratio assessment, and benefit risk assessment. That is a continuous process. prospective process, whatever the risk ratio, benefit risk ratio today, that could be changed tomorrow and day after tomorrow. So we need to be vigilant in that mode, and how we can use ethical artificial intelligence and of course the rational therapeutics. So a few important words for this future trial engine. It should be precise, predictive, inclusive, and trusted. So if we talk about data-rich trials to patient-relevant decision-making, that should be the goal of future generation trials. So I think with that, I think I can stop screen sharing here, and this was my take on that topic by Professor Brigadier Praful Mohan sir. We really miss his presence, but thank you all. And at the end, I want to thank Team Clarnet for giving us this digital platform to reach more than 550 delegates today. We are really thankful to all the delegates who were there, who were supporting us. I request you all who are not yet becoming the member of Indian Pharmacological Society, please become the member of our respected society, Indian Pharmacological Society. And may now request Team Clarnet to have their presentation, if any, for promotion of their platform. And we are really thankful to you all.
So, sir, we don't have any presentations as of now, but thank you so much to all the dignitaries present here. And with this, we are closing the session for today and looking forward to hosting you again, sir. Thank you so much. Have a good night.
Thank you very much. Thank you. Thank you all.